Good morning. It's now my pleasure to introduce our next participants, Dr. Iraj Ali, CEO, and Dr. Ed Samuel, EVP Technical Operations at Achilles Therapeutics. So the format for this session is approximately 25 minutes fireside chat, but we'll start off with some high-level overview slides of Achilles and its manufacturing capabilities before we move on to the Q&A portion. If you'd like to ask a question during the session, please enter it into the chat box in the Zoom window, and we'll try to ask them on your behalf. Raj or Ed, please begin.
Thanks, Julia. We really appreciate the opportunity, and thank you to Chardan for inviting us to the conference to speak a little bit more about Achilles. I'll draw people's attention to the disclaimer on the first page, and they can review that in their own time. So maybe just a few words about the company for those that might be not as familiar with the story. So we are a precision T-cell company developing therapies for multiple solid tumors, and really at the central tenet of what we're trying to do is to target a novel class of antigen in the solid tumor space, and that's the clonal neoantigen, which is a target present on all tumor cells and absent from healthy tissue, probably what most of us would describe as the ideal properties for a cancer target. And it has been difficult to find and difficult to do so on a routine basis, and that's why we've developed a proprietary AI platform, critically it's validated on real-world patient data. We produce a trackable precision T-cell therapy. We'll talk more about that and what we can do with that, and we believe take a differentiated approach to manufacturing. So we'll go through all of that with you in this panel, we hope. So very briefly, where are we today? We're a clinical stage company. 2 studies open and recruiting, one in non-small cell lung cancer, the Chiron study, one in melanoma thetis. Both have important readouts in the second half of this year, where we're looking at monotherapy data in the higher dose cohort, targeting doses of up to 100 million of the active ingredient CNET that we'll talk more about. But also in the melanoma study, we've opened the checkpoint combination arm, which we think will be an important and very significant arm of the study. We have a number of other solid tumor targets in development at earlier stages of development. So here, my last words really is to comment a little bit about the concept of the clonal neoantigen, which is steeped in the history of tumor evolution, which is a field that's really exploded in the last 10 years and really changed or a lot of what we think about cancers around. So we know that tumors are constantly evolving, and that has really been the central theme of why it's been so hard to treat them with targeted therapies, because genetic diversity not only it creates an unstable antigen environment, so tumors are able to gain function and to lose target, and that makes it really difficult when you're targeting a single agent. But this very basis of the way the tumor behaves is also a weakness that we exploit when we develop drugs, and that's cancer's Achilles heel and the derivation of the naming of Achilles. We know that there are a set of antigens or mutations formed early in the tumor's evolution, depicted as red on these diagrams, that are present throughout the tumor's life. And those are what we can target, the clonal neoantigen, at the trunk of cancer's phylogenetic tree. So while we really struggle with this challenge of heterogeneity, genetic heterogeneity in tumors, it's the clonal neoantigen that unlocks the potential for long-term efficacy in these diseases. And we've developed unique technology to identify clonal neoantigens and target them. And we target multiple clonal neoantigens, which we think gives us the best chance of long-term durable disease control, even in late-stage metastatic cancers.
If I just take you through at a very high level the manufacturing process itself, so you can see here, patient comes into surgery on the left-hand side. We perform a tumor resection, so a piece of tumor along with a blood sample from the patient are then shipped to our manufacturing site. And these represent the kind of starting materials for manufacturing our precision T-cell therapies or clonal neoantigen T-cells. So really importantly, from that tumor, we're able to take regions from the tumor that are sent out for genomic analysis and sequencing along with a germline blood sample. And that enables us to identify each patient's clonal neantigens that are specific to them. And so that's the kind of genomic element that runs in the middle here. And it's that set of sequencing data from the patient's tumor that enables us to generate a patient-specific list of clonal neantigen peptides that are used in the manufacturing process. So, whilst that piece is ongoing with the tumor, we're able to isolate the tumor-infiltrating lymphocytes, and from the blood we generate monocyte-derived dendritic cells. These both represent kind of our intermediate products that are frozen down while we wait for that genomic analysis to take place, and the sequencing data to run through our bioinformatics platform, Peleus. The point at which we have the patient-specific peptides in hand, the TILs and the DCs, we can then bring them together in a co-culture shown here on the kind of right-hand side, whereby the DCs take up the peptides and they present them back to the T cells, and we're driving the selective expansion of those clonal neantigen T cells. So for us, TIL represents the starting material. We're interested in a subset of those TIL, the clonal neantigen-specific T cells that are expanded in that co-culture. And then we freeze that final drug product down after harvesting, and that gives us the flexibility to ship to clinical sites for reinfusion. So really importantly, one of the kind of key principles at Achilles is to be able to determine the active component of our drug product. So that's the active clonal neantigen T cells. And so that's importantly, firstly, for being able to quantify the dose. And we're able to do this through first being able to prospectively identify those clonal neantigens from the patient's tumor cell, and then in a number of assays that we have, be able to re-stimulate a aliquot of the patient's product, and that enables us to determine what the active component is, and we do this by measuring cytokines, interferon gamma and TNF alpha. So once we know what the active drug component is, we can then track that in the patient. And so that's really obviously incredibly important and powerful for being able to determine the expansion of those CNETs in the patient and then track them over time. So being able to correlate both the dose with the persistence of those CNETs in the patient is critical for understanding mechanism of action and characterizing our drug products. So this slide here just talks to one of the key concepts at Achilles, and this is a manufacturing summit. And at the heart of Achilles from inception was really understanding how we both close the manufacturing process and deliver at scale. And so what you're seeing here is one of our key projects that closes the process from the point of surgery. So we've developed a bespoke tumor collection processing device on the left-hand side here, or collection device, and a processing piece of kit that sits in our GMP clean rooms. And so once that tumor goes into the pot on the left-hand side, the process is then closed. The tumor can be shipped back to the manufacturing site, where it's run through our closed tumor processing device. And this really is a key driver from taking what is a very complex and operator-dependent process down to something that can be done in a kind of matter of minutes in 1 or 2 steps, and a really key project for us to be able to deliver at scale. And so lastly, just kind of talking you through the kind of the manufacturing footprint that we have at Achilles. And so the Royal Free Hospital is currently our site for manufacturing our first-in-human products for both that Chiron and Thetis studies. And we've been manufacturing there since the inception of those studies in 2019. And most recently, we have just bringing online our second manufacturing site in the UK at the Cell and Gene Therapy Catapult in Stevenage. And this represents the first scale up in our dose capacity at Achilles. And so looking to kind of bring clinical manufacturing online at the Catapult later this year. With regards to the US as well, establishing our footprint at the Centre for Breakthrough Medicines is really a key component to being able to deliver globally for those first-in-human and registrational studies as we begin to establish capability there, and remove some of the logistics and complexity of shipping starting materials and drug products globally. Clearly, for us, ownership of the entire manufacturing process, all under one roof, is a critical component at Achilles. And so, on the right-hand side of this slide here, you can see one of our kind of end goal, which is developing and building our own facility in Hayes in West London in the UK. And so, looking to bring that online to develop thousands of patient products over the coming years. So I'll just hand back to Iraj.
So as mentioned, we think there's an exciting series of catalysts this year for the company and exciting data that we want to mature. We're moving into a higher dose cohort of patients on process 2. Looking at the monotherapy data, we expect to bring out sort of 5 to 10 patients by the end of the year and try and sort of squeeze in an update in Q3 if possible. We'll also be looking at a combination with checkpoint inhibitor. More difficult to predict exactly how that matures, but we would expect to have 1 or 2 patients by the end of the year. Ed's talked to the maturation of the manufacturing footprint, and we can talk about tap through the questions if that's helpful.
Great, thanks for that overview. So to drill down on some of the issues that you covered, so you mentioned that you use TILs as the starting material and then incorporate a genomics and bioinformatics workflow to identify the clonal neoantigen targets there. Can you expand a little bit on how your bioinformatics platform sets you apart from other approaches, including other TILs or cell therapy strategies within the space?
Yeah, absolutely. I think that's really central to what we're doing that's different. There are a lot of people out there that are looking at neoantigens, not many looking at clonal neoantigens. What we've learned in the field over the last, I'd say 5 to 10 years about identification or assessment of genomic material is twofold. So the first is it's computationally extremely complex, much probably more complex than many of us had envisaged back in the early days. And it requires validation, which is a consequence of its complexity. So at the center of what we're doing at Achilles is we have exclusive license to TracerX data, and that allows us essentially to validate what we're doing and gives us an amazing starting point. That's the largest and probably most significant data set in the bioinformatics field. It has about 800 patients recruited in non-small cell lung cancer, tracked over 5 years. We're into 3 to 3.5 trillion different reads, 3,200 separate tumor samples, all annotated informatically, and have the phylogenetic tree of the cancer developed. So it's an unparalleled data source that allows us to truly define what clonality is. And that's what we've used at the center of our platform, Peleus. And then more or equally important is it allows us to validate what we're doing with real-world patient material. Because otherwise, I think there is a danger that whatever your way of identifying neoantigens, whether that's computational or empirical, it needs to be validated. You need to show that these are real antigens, they really create T-cell reactivities, they are immunogenic, and most importantly, they're actually present in patients because we're using the patient's own immune system. So I think it falls down into 2 broad areas. One is computationally, we believe our approach is probably the most advanced out there. We've developed now a very complex AI platform that solves the heterogeneity problem from a computational basis. And secondarily, we have validated our calls with real-world data, both from TracerX and what we do prospectively when we create products every time we manufacture them. So we think that our approach to identifying new antigens is unique and leading in the field.
Great. And then with regard to cell therapies, you talked about your approaches to closing the system and reducing, variability associated with human operators, et cetera. I think one of the other factors that has been a challenge in the autologous cell therapy space is reducing vein-to-vein time. Can you discuss how you're approaching those type of issues and whether there's elements of your manufacturing process that can help overcome those additional challenges relating to the administration of your cell therapy?
Yeah, happy to pick that one up. And I think, reducing vein-to-vein time is, clearly an important topic at Achilles. But I think, perhaps less so than, say, in the space of CAR T therapies, where the patient is waiting for a product. With regards to our CNET products, generally we're collecting material at surgery, and patients are then going on to other treatment therapies while they wait for our product to be made. But nevertheless, it is still a very important driver to be able to kind of compress some of the timelines. And we see, the establishment of the manufacturing footprint and bringing in-house capabilities, whether that's, the ability to make our own personalized peptides for the patients, as key drivers to compressing vein-to-vein time. I also think, some of the kind of maturing platforms that we're seeing with regards to electronic systems and digitizing patient record or patient batch manufacturing records will contribute to being able to, expedite things like document reviews and QC and QP time. So I think in part it will be a combination of 3, 4, 5 different things that will kind of reduce that vein-to-vein time. And we're going to see the kind of advancement of, I guess, of things like sequencing times come down as well in the future. So it's something that we continue to look at very closely, and with a target of getting kind of to circa 6 to 8 weeks for a commercial product.
Got it. Great. And so you also mentioned the process that you're using, Velos. You're currently at the second generation process. Can you expand on the differences from the first gen process and what lessons you've learned from advancing to the second generation that you're currently at?
Yeah, so I guess, maybe just talking to the kind of differences at a high level between what we've presented previously on our process 1 and the new process that we've just bringing into clinic, process 2. So, we've looked very hard at kind of optimizing things like the cytokine feeding regime that we use in both the upfront expansion of those or isolation of the TIL intermediate, and also carrying that through into our patient co-cultures. Still retaining the DC specific expansion, which is key to being able to drive the expansion of those CNETs, but then looking at kind of a non-specific activation, polyclonal activation of those T cells to get a boost of the final product. And I think what's driven us towards that is, we've obviously collected data with our process 1, and the intent now is to kind of move up the ladder with regards to CNET dose, and we see the optimization of both, the manufacturing process with regards to, the culture as key to being able to increase that dose. I think clearly, just the virtue of being in the clinic and collecting real-world patient data from our current thetis patients is important for understanding things like tumor biology, TIL yields, and collecting that genomics and on the antigen data, which all contribute to the kind of iteration of the manufacturing process that you see today.
Got it. Great. And so in terms of analyzing the final TIL product, I think this is an area that's garnered a lot of attention within the cell therapy space, as you alluded to earlier as well. So can you discuss a little bit about the safety and potency parameters that you're looking for within your final TIL product and the characterization that you do?
Yeah, no, absolutely. And I guess one of the key differentiators perhaps for the Achilles products are that they're cryopreserved, and that enables us to perform the full scope of required release testing prior to patients being infused. And so I think, the safety assays are very well understood by the field. So, looking at sterility, endotoxin and mycoplasma testing, that we've now kind of, or ahead of clinical trials, fully validated those assays to support the clinical studies that we're in today. I think that perhaps the more interesting kind of evolution is looking at things like potency assays and beginning to understand how do you characterize your product? What is it the regulators want to see with regards to the kind of matrix of assays that you can support both understanding, mechanism of action, but also trying to simplify the assays that you have to use in a GMP environment to release products. And so, we've talked a little bit about our ability to be able to identify the CNETs through using those patient-specific peptides. So our potency assay it enables us to use the same peptides in manufacturing, to be able to use in an assay to identify those CNETs and tracking them in the patient. But I think more broadly, you know, it's collecting data on outside of the kind of GMP and releasing product space, and understanding, you know, what do things like CD4 and CD8 ratios look like? How are we collecting data on the fitness profile and the phenotype of our products? And we know that regulators want to see a much broader data set as you move through the clinical development kind of roadmap to getting to a commercial product. And so we've had to think very, very early at Achilles about what a potency assay would look like, and that's something we continue to do, you know, pretty regularly, both in R&D and our GMP space.
Got it. So thinking about the you know, comparison of the Velos II process versus, let's say, a standard unselected TIL approach, how do you think the number of active cells compares with what we know about, let's say, a standard TIL process that produced billions of cells but with uncharacterized specificities?
Maybe I'll pick that one up. So I think the short answer is we don't fully know the answer to that. What we know is that an unselected TIL process, only a subset of the cells are tumor reactive. Now, the estimate of that percentage can vary quite significantly depending on the method that was used to determine it. So we have an issue of not having a sophisticated assay there. What we think, as best we can, looking at what we feel is some of the most appropriate techniques, is it's somewhere between 2 to 5%, and maybe at the very highest it might be 10%. But the published data would suggest, and certainly data in our hands and experience, although we only look at clonals, suggest that somewhere between 2 to 5% of a TIL product is actually active, tumor reactive, and certainly at the lower end for clonally reactive. So I think that shows you that the majority, or would suggest that the majority, is not tumor reactive, or not binding to a neoantigen, at least. And I think probably the proof of that pudding has always been in the eating, which is, you know, if you follow the TIL data over the last 20 years, we've never seen a convincing correlation between total T cell dose and clinical response, whether that be ORs or CRs or PRs or OS. It's never correlated convincingly to TIL dose. So that data has been around for a very long time. I think it's probably come more into the forefront in the last few years as the FDA's sort of put their position out, that lack of correlation is probably not sufficient for potency, because it would suggest that T cell is not the sort of specific reagent of your product that is doing the disease amelioration.
Got it. That makes sense. And then I think more generally, are there other innovations that Achilles is exploring that could expand the scope or the capabilities of its platform? And related to that, are there areas where that make Achilles' manufacturing platform stand apart from its peers that we haven't already discussed?
Yeah, that's a great question. I genuinely believe that we're in a very, very exciting time in the field of cell therapies, and specifically T cell therapies. There is a huge amount of technology that can be applied that expands the scope. We're currently looking at, I think very, very rightly, at T cells that are sourced from TIL or the tumor mass. There is also now increasing data that there are other sources of T cells that are going to be important to look at, like blood. So we think that there is different versions of essentially the same product that could be more amenable to different disease indications. And also, I think that the natural place that everyone is looking at is looking at gene editing or gene knockdown or various ways of adjusting the DNA of the T cells that you're affecting to knock out or knock in different activities to increase the anti-tumor effect and potency and fitness. And I think that, yeah, I think that the companies like Achilles have a very significant advantage, because you can characterize your tumor reactive component. And that's really what matters. If you have a product that is, essentially 95% not tumor reactive, and then you gene edit it, I'm not sure what that means for you. What you really want to know is, what is the effect you've had on the tumor reactive component, and can you track that in the blood, and is that doing what you want it to do? So I think we have a very significant advantage when you think about taking products like these to the next level.
Fantastic. So I see we're at the end of our 25 minutes. So I'd like to thank you guys again for the great discussion and for your participation. And we'll see you tomorrow for the cell therapy panel as well. And we'll discuss some of these issues in more detail.
Thank you so much for the time. Yeah.